Hello, everyone. Is my audio clear? Okay. So we're going to start a new lesson. And in the last lesson, previously, we started off with chemistry of nucleic acids and the chemical nature of genetic information. And then we discussed about replication of genetic information. So I understand we all our syllabus starts with Mendel's laws, but I think once we get to know the nature, replication, and then the important one to know is about the genome structure, and hopefully then we will be able to make full better understanding of Mendel's laws and so on. So we are going to start the chapter on genomic structure, chromatin, and nucleosomes. In this particular chapter, we will learn types of sequences, right? And how diverse they can be, how many different types of sequences one can have. Then we'll also have a discussion on how chromosomes duplicate and segregate to daughter cells. And then we will discuss about the nucleosome and higher order chromatin structure. We'll briefly touch upon regulation of chromatin structure. But otherwise, the chapter in the textbook has a lot of details. We don't have to cover all of them, but still I think we will cover a significant portion. If you have bought the book and are studying along with me, I can assure you that we will finish a large portion of the critical points or components of the textbook. And that way, maybe by next semester or sometime, I hope you will be able to cover the whole book, and that will empower your knowledge, understanding, and intelligence in terms of genes and genomes. Okay. So the first thing, we need to also have a bit discussion about several terms. In this class, I will focus on different terms so that you will get used to those terms and you will be able to understand when I use them properly next time. So we know that there are genetic information is made up of DNA, at least in all cellular organisms. And then what is a chromosome, right? Chromosome is not nothing different. It still deals with the genetic information. Only point is, as an entity, this DNA or the whole DNA is bound with along with proteins, okay, and or it is coated with several proteins, and that we refer to as a chromosome. So one organism can have several chromosomes. Say, for example, we have in our cells, humans have 46 chromosomes, right? I think you all know that it is 23 pairs, right? 46 chromosomes. Each chromosome, at least in eukaryotes, is a linear DNA, right? Linear DNA molecule, which might be coated with a lot of proteins. So there are some things about that we will have to understand, I think, as we will see in the next slides. But first thing, we also have to appreciate that DNA should be packaged. In the next classes, we will also do a lot of questions or problems based on DNA length and other things. As we progress, the complexity of the problems will also increase. If I didn't mention, all of CAA, if they were to be conducted, they will be only based on calculations and thinking. You will not have so much writing to do. It's more of diagrams and calculations. So all the DNA that is present in one cell should be passed on consistently to the daughter cells, right? And for that you need the DNA to fit inside the cell. The DNA should be protected, and then the DNA should be transmitted to the daughter cells, and so on. And whatever genetic information that is present in the DNA, okay, if you recollect from what we were discussing in the previous class, DNA is just a chemical, it's matter. When you say genetic information that has some sequence, as we progress in this course, we will get to those things. So the genetic information that is present in the DNA should give rise to a protein, for example. And for that also, you need several proteins to be associated with the DNA. Okay. Here is what I would want to discuss is all the chromatin or chromosome. It's kind of almost the same, DNA plus proteins. And we are going to learn a little bit about proteins today, what proteins are associated with the DNA in our chromosomes. They are called as histones. Okay, this is about, when we discuss in this chapter, majority of the things we are discussing is about eukaryotes. Okay, because they have a higher order or complex chromosomes compared to that of bacteria. Bacteria or prokaryotes, they have very simple simple genomes. In the assignment, I noticed that many people have put in a lot of efforts trying to find. At least some people have done a very good job with that. And there are also several other non-histone proteins. Histone proteins are something like we will study in the fourth class on this particular lesson. But this is the the red line that I have drawn is supposedly the DNA, and these are assumed that this is the nucleosome, or this whole structure is called as nucleosome, and the green ones are the histones. Histones are in a way non-specific, non-sequence specific, okay? Non-sequence specific DNA binding proteins, okay? That means what is meant by sequence specific and what is meant by non-sequence specific. When you say sequence specific, they would have wanted, say for example, a DAM methylase that we have discussed in the last class. It is sequence specific. It can only bind and act upon this kind of sequence. It cannot act on every DNA, right? Whereas nucleosome, if there is a DNA and you put in nucleosomes along with it, it will go and bind to the DNA molecule. So that is sequence non-specific. If you also think of it in a different way, what I want you to appreciate is assume this is the phosphate sugar backbone, right? And you have bases here. So I'll just put it like this. Okay, these are, assume that there are bases here and they are bound by hydrogen bondings. If it is sequence specific, if a protein is sequence specific, this I'm telling you just for thinking purpose. It is, if a protein is sequence specific, somehow it should make interactions with the bases. That is how, I think you remember minor groove and major groove, where some of the groups of the bases are exposed, to which using which the proteins can bind and determine if they are binding in sequence specificity or not. Whereas non-sequence specific binding proteins, they would have only interacted with the phosphate backbone. They don't care about what is the sequence and so on. They would have made majority of the interactions with the deoxyribose and the electronegative phosphate groups. Okay, that is how one has to think about interactions between any of the molecules, especially with regard to DNA. And sequence specific should interact with the bases. Non-sequence specific ones don't have to interact with the sequence with the bases. They just have to interact with the backbone. Okay. There is one important thing that we have to remember is each of our cells, human cells, they have about it's a haploid set. What is a haploid set? We have about, we said 23 pairs. That means we have something like this. Assume that in our gametes there would have been only 23 chromosomes. That is haploid. And because when it is in pairs, that is called as diploid. Okay, as a haploid set, we have about 3 into 10 to the power 9 base pairs. That's huge amount of DNA. And if you consider it for a diploid set, right, it would be 2 meters. In one cell, we are talking about just one cell. You put all the chromosomes, I'll erase these. If you put all the chromosomes in end to end, chromosome 1, 2, like this, then the total length of the DNA would be about 2 meters. What is the, shall we make a quick calculation? So 6 into 10 to the power 9 base pairs. That means if you say, how many bases would that be? 1 turn of DNA, right, is about 34 angstroms or 3.4 nanometers. I'm not going to do the calculations for you, but I would want you to appreciate how this is. Say 1 turn has about 10 bases. So you can calculate 10 to the power 6 into 10 to the power 8, and then multiply it with 34 angstroms. You can choose whatever you want to do it, and then you convert that into meters. Okay, so what I want you to appreciate is DNA from one cell, when put end to end, is about 2 meters. It is taller than majority of us, right? It is longer than majority of our whole body. And yet, so assume that this is us, it is longer than us, and the cell is, or the nucleus within the cell is 10 to the 15 micrometers. Okay, I hope you can see the how that long DNA should be put in a very, very, very tiny molecule, tiny component that is not visible to us, our naked eye. So that is called compaction. You take a large DNA molecule, fold it upon itself, use supercoiling, fold it further rounds, and then be able to package into it. And that is one of the, everything starts with nucleosomes, like I discussed a little bit earlier. And if you have really folded the DNA or compacted it so much, how is it possible that it can still give rise code for the proteins that it must, right? The expression of genetic information. So this lesson is about it. How is it packed so tightly and put into a component like this? And yet, how is it accessible for a gene expression? Okay. And remember that the focus will be about cell division and many other things like that. How does a genome behave? So in different organisms, the shape and number of the genome is different, or chromosomes. So in bacteria, they are almost all bacteria, it is circular. And in majority of the other organisms, eukaryotes especially, it is linear. Okay, I think we discussed about it when we were talking about replication. Circular is something like this, which is very stable, whereas linear one, we discussed one of the key points or the troubles with it. It's called as end replication problem, and we have also seen in the last class how nature solves it in the form of by synthesizing telomeres, by telomerase. And we also have discussed about the priming protein, which is an innovation of the by the viruses. And in bacteria and other things, usually it is circular DNA. but there are bacteria which have two circular DNAs as well as a part of genome. And they can be single or numerous. Numerous in, say, as we were discussing about our own chromosomes, we have 23 pairs of chromosomes. That's a large number. And there are other organisms which have even larger ones. So prokaryotes, circular, eukaryotes, linear, and in prokaryotes, majority of them one, but in eukaryotes, usually it is more than one. And in prokaryotes, they have plasmids. Plasmid is not a part of the bacterial genome, okay? You can consider them as molecular parasites, very similar to that of viruses, right? Viruses, what are they? They go onto a cell, they will infect the cell, and then they will make more copies of them, and then they kill the cell and get released out. Plasmids have almost a similar one. They enter into the cell, or they are taken up by the cell, bacterium, and they may confer, or what, they will just live along with them and keep replicating along with the cell. They don't really kill the cell directly, but they use up the nutrition and all those things. They may occasionally confer some advantages to the cell, but still they are independent entities. Yeah, we discussed about the challenges with circular or linear as we were talking here. In circular, they are much more stable. There is no problem of end replication problem. In linear, the problem is the end replication problem is there, and there is also a problem of exonuclease activity, because of which they might get degraded from the ends. So these are solved by synthesis of telomeres by telomerases. So let's see a table. Whenever you come across a table within your textbook, please don't leave it. Try to have a look at it and try to analyze it a little bit. In prokaryotes, Mycoplasma E. coli, Escherichia coli, that is the one popular one. You see that they are having single or one chromosome each, and copy number is also each. When you say copy number here, it's about whether it is haploid or diploid. And they are all circular. In all of the prokaryotes that you see, you'll see that their majority of them are circular, and occasionally you may have something like one linear and so on. And if you take eukaryotes, you may have multiple numbers. You don't find one anywhere. You have 16 more than 3. Saccharomyces cerevisiae, yeast, that is what we have discussed a few in the last class. And also here, they have 3 and 16 like that. Okay, they are more in number. And copy number is something important. It's 1 or 2. These two Saccharomyces, these are fungi. Fungi can exist as hyphae or monogametic, in a gametic stage or in a diploid stage. So at half part of the life cycle, it might remain as in a haploid state, and at some point it will remain as a diploid state. In humans also, we can have something similar, okay? But our haploid state is very, very small. It's only in the stage of gamete. And gamete doesn't do any functions other than fusion, and then zygote forms. And from then on, a life has started. But for them, in yeast or fungi, you can see that it exists as a haploid for a long time, and then it might become diploid and so on. It can still absorb energy and nutrition from the outside and grow. That's why they put it as 1 and 2. But in our case, where in humans, our gametes are haploid, but they don't do any absorption of energy and other things. Okay. So there are some more things I would want you to understand. Tetrahymna, Thermophilus, it's a protozoan. It has micronucleus, 5 chromosomes, but in macronucleus you have 225 chromosomes. They have two different nuclei. One is micro and one is macronucleus. And you should see the copy numbers, that you see that there are very large number of copies. And here is the genome size. So I hope you understood that from the table. You can see the diversity of how genomes is, and they can have a wide variety in the number, copy numbers, and also in the genome size. So I hope we will discuss a few words before we get into it. What is a nucleoid? Anybody? Can anyone guess? You must have heard of it. In prokaryotes, the DNA, they do not have nucleus, but their DNA is still coated with, or the chromosome is coated with some proteins, and whatever exists there, that is referred to as the nucleoid. Okay, prokaryotic genetic material or prokaryotic chromatin can be referred to as a nucleoid. Diploid is 2n, and haploid is n, what we have discussed about. Set, if it has only one set, then it is haploid. If it has two sets, same copies, okay, pairs, then it is diploid, and you also have to get used to n and 2n like that. What are homologs? Say, for example, a cell has a chromosome A, and chromosome B, and chromosome C. If that particular cell is diploid, then it would have another copy of A, another copy of B, and another copy of C. So which two are homologs? A and B are not homologs, whereas this A and this A are homologs. That means they have same sequence. Okay, chromosomes that have same sequence like this, A and say I'll put A prime to differentiate, A prime are homologs. Here B and B prime are homologs, and C and C prime are homologs. Okay, and polyploidy is something. In some cases, I hope you remember, if you have studied in botany in your +2, you would have seen that endosperm is in 3n. It has 3 sets because of double fertilization during fertilization in plants. You will have 3n, which because of which now you actually have 3 sets of n, and that will be called as triploid. In some cases, they may have 4n or 8n. It usually will be in multiples of 2, except in cases of endosperm and so on, or 16n also. Sometimes genome gets duplicated and will result in multiple copies. What is the normal one? It's always 2n. 2n is normal, but because of some events, it might become 4n, 8n, and so on. And that is whole genome duplications. They are referred to as polyploids. Okay, and gene dosage is number of copies of a gene, of a particular gene. Okay, we are not talking about the whole genome set. We are only talking about one gene. Say, for example, there are 2 chromosomes. I'll write chromosome A and the homologous chromosome, chromosome A prime, and assume that there is a gene called X. Okay, and A prime is homologous, and it should have one X chromosome as well. But because of some genetic activity or mutation, it doesn't have this. Okay, so what is the gene dosage? It's only 1 copy of X. So when you're trying to figure out which are present and which were not present, that is where of particular genes, you're talking about simple genes. So remember it as gene dosage. And here we will discuss something about megakaryocytes. Those are blood cells which produce platelets. Our body or in the blood, you need lakhs of platelets, and platelets are involved in several aspects of forming blood clots and also eventually dissolving blood clots as well. To produce that, there is a the cell that what we refer to as megakaryocyte. Mega is for big, megakaryo is nucleus. That means it supposedly has very large nucleus. Why does it have large nucleus? Because it has 28 copies, 128 copies of each chromosome. That's, you can say it as a kind of polyploid as well. And it will, once it matures, it will fragment into several thousands of platelets. Okay, this is a nice example of trying to know in our own body we have cells that have 128 copies of each chromosome. We have gametes. Gametes are haploid, that is N, that is haploid. Most of our other cells, majority of our other cells are 2N, they are diploid. And we have megakaryocyte, which is having 128 copies of each, and that is kind of polyploid. Usually polyploid, when we say, it means all these cells in the body are. But here we can just still take it as an example, as representation of polyploiding. I think I also gave this as a question to find the differences, similarities and differences between prokaryotes and eukaryotes as assignment. I noticed that some people have done fantastic about it. So bacteria are haploid, and majority of the eukaryotes are diploid, but they don't mean some of them can exist as haploid as well. I hope you notice that in this case you have 2 orange, 2 blue, and 2 purple, right? The 2 purple ones are homologous chromosomes. Whereas in the haploid cell, you only have one copy of each of these chromosomes. The other differences would be that membrane systems are not present in prokaryotes. There is no nuclear membrane. The whole DNA is called as the nucleoid, and they may have a flagella and other things. It is very simple. Whereas in eukaryotes, you have mitochondria, endoplasmic reticulum, Golgi complex, and many other complex cell organelles are there in eukaryotes. What we should see also is about the genome size and organism complexity. We do consider prokaryotes as very simple organisms, right? Because you don't find as much complexity as we saw in the last slide. They are very simple. Nothing, just acquire energy, multiply. Nothing else is that. That is what every other propagating entity does. But still, there is a level of complexity. If you see prokaryotes, or majority of the times they are single-celled unicellular, right? They are unicellular organisms, whereas eukaryotes, except from protozoa, majority of them are multicellular organisms. That means they form groups together, and then they have a degree of differentiation. We will talk about differentiation and other things at a later time. Or simply to put is that if you say we have cellular differentiation, can you imagine that we have about something like 210 different cell types in our body? And when you say cell type, neurons are in a different shape, morphology, and function. And you can take a skin cell, which may be different. You can take the epithelium of our intestine. It has a different shape and function. The total genome component, the content of the genetic content of in the neuron or in the epithelial cells or in the intestinal epithelia, the genome is same, yet they have highly different form, morphology, and function. And those cells, the process of forming different types of cells is differentiation. Okay. If anybody wants to say an organism is multicellular, then they should show that there are at least two different cell types, whereas the genome should be same in both the cell types, but at least they should be different form and function. Differentiation is key to multicellularity. Okay, so from here to here, we are increasing in complexity. What does that mean? Organismal complexity is increasing. So if you see that in that sense, genome size, it's increasing to a very high rate, right? And number of genes is also increasing. Something like E. coli has about 4,500, and something like humans about 25,000. I think it's updated now. It should be somewhere around, say, 20,000. That's fine. And you have some of the plants. They are huge. Some of them are polyploids, and they have large number of genes compared to us. Okay. It is likely that we have a different lifestyle. Animals have a different lifestyle. We can move around and get things done. But plants, they cannot move around. They be where they are and yet achieve what they should. That is to acquire energy and to reproduce, right? So probably that's one of the reasons why they may be having more number of genes. We have less number relatively, but still we can accomplish many things with those genes because we have capacities of moving and so on. Okay. And something called as gene density. Say, for example, I would like to describe what is gene density. Here is something like this. This is a DNA, and assume that it's double-stranded. There is another DNA molecule like this. So if there is assume these are each one of the rectangles I'm drawing are representative of genes, right? With the same length of DNA, I'll give it this as state A and state B. Which of these has more number of genes within the unit of DNA? A has more number of genes within the same unit of DNA. So A has high gene density, genes by MB of DNA. Whereas in B here, you do not have as many genes. So the density is not very high. If you look at some of the cells, the gene density is very high in the prokaryotes or simpler organisms. As the organisms are increasing in complexity, genome size is increasing, right? And but the gene density is actually reducing. It's not so much. I hope you would recommend, you could understand that, say, you have 4.6 MB of genome in E. coli, and it has 4,400. Say, if I need to have somewhere around something like 20,000, what is the shortest length that is possible using this as a calculator? Yeah, you will need 46, say 4 times of it, right? So somewhere around, it should be about 20 MB or so, right? Approximately, just do it. But look at the size of our genome. We have 3,200 MB of DNA. That means the gene density is very less. There is a lot of DNA that is actually not coding for any genes. So I want you to appreciate that part. So realize that we have very large amount of DNA, but majority of it is not so useful, or at least within our perspective or the current understanding. We don't know if there is any function to the rest of the DNA. So I will want to use this slide to talk about compaction, because we have to talk about viruses actually. Say this is a DNA, and I'm going to draw I'm going to put a smaller DNA molecule or genes, okay? Say, for example, a gene starts here. Okay, sorry, it doesn't work that way. I have to draw it before. Yes. So if I am going to draw a gene, I will draw a gene like this. I'm calling, typically you would have a gene like this. It is a double-stranded DNA, right? Okay, I'll put it like this. Double-stranded DNA, one gene can be coded here, and the other one is here. If some of you are having trouble, what is a gene? Is it sitting on the DNA or so? Don't have any rigid mental images as such. Just leave it fluid and be open. We will discuss as we progress. We will know it. I'm going to draw another DNA molecule. Same way. Only thing, I'm going to have a little bit of difference to it. So in the first one here, a gene has started at this location, ends here. This is the start for the second gene, and it ends here. Some of the cases may be that a gene starts here and ends here, whereas the second gene may start here and end here. So there is a overlap. Those are referred to as overlapping genes, or ORF is for open reading frame. In the next classes, we will know what is an open reading frame. For the time being, you should know that open reading frame produces or codes for a protein. Okay, that means this one is an ORF. That means if it is transcribed and translated, it will produce a protein. The same is the case with this one too. Okay, here this open reading frame, there are two open reading frames. One starts with this and the other one is somewhere here. When this part of it reads, and it will produce a protein, something that might look like this. And when this one is expressed, it might produce a protein, something like this. Okay, but these two are overlapped. That is meant by overlapping open reading frames or genes. The third one is something little bit different. You have a DNA molecule like this, and there could be a open reading frame like this. One strand of it is coding, has a open reading frame, so it can produce a protein that might look, say, something like this. The other strand can code for a different protein, which would be read in this direction. We will discuss those in the later time. It's a good time to introduce these terms to you, so you will feel comfortable. And the second open reading frame is producing a different type of protein. So this is on the reverse strand. One strand has a gene, and the other strand has another gene, like this. So they both produce, there is an overlap, but they are on different strands. As an open reading frame, they are not overlapping, but as a part of DNA, you can assume that they are kind of overlapping. I hope you understood what is overlapping open reading frames and what does it mean to code for a DNA. Every time we say a gene or a genetic information, remember that it is a particular given sequence of DNA, right? GATC, AC, GG, or whatever it is, that sequence itself is the information. Some sequences code for RNA, some sequences code for protein. Those sequences that code for protein are called as open reading frames. Okay. So here is some of the comparison of chromosomal gene density. And if I forgot to mention, again, in viruses have very high gene density. And that is because they have a large number of overlapping genes. When time comes, we will discuss about the genome of the coronavirus, so that it should be that we will understand it much better. Here is comparison of chromosomal gene density about E. coli, yeast, Drosophila, that is the fly, and then humans. The green ones show the genes, the actual genes that code. And I hope you know from your last year course in biology for engineers, you know what are introns. And there are certain regions that are highly repeated sequences. And here is the RNA polymerase gene, and then we have intergenic sequences. I think I have to describe what is each of them before we further go ahead. So this is a gene, right? Like we discussed in the previous slide. And some of the genes are split, right? With which are the unshaded ones are exons, and these are referred to as introns. Those are intervening sequences. They are sequences that will be removed out during splicing, RNA splicing. And then I want you to understand what is intergenetic sequences. Say, for example, this is gene 1, and there is another gene here, and which may have its own introns. The space between them is called intergenic sequences. Inter, that means between two genes. That is intergenic genetic sequences. Okay. Repeated sequences we will see in the next class. So if you see E. coli, it's all shaded in green. Green are all genes. Whereas with Saccharomyces, you can still see that their majority of it is covered in the same thing. But when it comes to Drosophila, you see that the green parts have become very sparse. They are highly distributed. And in humans, when it comes to humans, you can see that it is much more uh, sparse, or green is very, very little that can be seen within this region. Compare the size of RNA polymerase alone. RNA polymerase in bacteria doesn't have, prokaryotes do not have introns in their genes. Saccharomyces also doesn't have, very little if there are, very few. So the size of the RNA polymerase has increased a little bit. And when it comes to the size of in Drosophila, it has much more, and that is because of the introns. Look at the size of the RNA polymerase that starts here and ends here, and it is very it is split into um, several exons. All the orange dots that you're seeing here, the orange ones here, are the exons. The remaining are all introns. So see the total size of the RNA polymerase gene. It was this much representatively, but whereas in humans it is much more spread out, split, and so on. Okay, that's why the genome density is usually less. I also want to have one more sequence that I missed, is regulatory sequence. If a gene is present, gene 2 here, and it should be expressed, that means it should be transcribed to start with, then you need a promoter, right? Promoter is the region which is recognized by RNA polymerase. It binds to that region and produces RNA. And then this RNA will be used by ribosome to produce a protein. The first step is being able to transcribe that gene. And for any transcription to happen, there should be a promoter. There are also additional sequences like enhancers and other things. All those sequences that are involved in the expression of that particular gene come under regulatory sequences. Okay. I'll take this and I will stop. Okay. I hope you remember about RNA splicing. What does the word splice mean? Is it joining or cutting? Splicing, if you see it, the meaning of it should be joining. Okay, I hope you remember. Let's go by this. The arrow mark here, it is shown, that is promoter. It is a kind of regulatory sequence. You can think of it. It is responsible or required for transcription of this particular gene. And in this eukaryotic gene, why am I calling it as eukaryotic gene? How do I know it? There are introns. Introns are only present in eukaryotes. And this particular gene has 3 exons: exon 1, exon 2, and exon 3. There is intron 1 and intron 2. So we need to get rid of those as we progress in gene expression. Upon transcription, a RNA molecule is produced, which is referred to as primary RNA transcript. And that is spliced, and eventually, or you call it as mature mRNA, which means to say that it is ready for export into the cytoplasm, and then translation would happen there. In eukaryotes, replication and transcription happen within nucleus. Translation happens within cytoplasm. In prokaryotes, everything is together because there is nothing like nucleus. So upon transcription and maturation of mRNA should get rid of these, right? I will ask, it's a nice way of referring, making your own statements, like, is splicing removal of introns or attaching or binding of exons or joining of exons, right? There are two statements: removal of introns or joining of exons. What is it? Removal of introns doesn't say, it says that these introns are removed, but it doesn't say anything about what happens to the exons, right? But if you say joining of exons, that means you say this is exon 1, 2, and 3. These are exons are joined, which would only happen if introns are removed. So RNA splicing is joining of exons. Like that, if you can reflect on whatever things that you're learning and try to make a statement for yourselves such a way that you will be able to remember things for a longer time, then you will be accumulating knowledge much better and faster. Okay, yeah, this is the last slide, and we already have discussed the same thing. And you have to know about the introns and repeated sequences in the genomes. Gene density um, reduces as the complexity increases. The size of the introns, or the number of introns, average number of introns, is also increasing, right? At least in this case. And the percent of repetitive DNA. We have a large amount of repetitive DNA that we will discuss in the what is meant by repetitive DNA. We will study about true chromosome structure in the later classes in terms of centromeres and so on. But otherwise, I think you have seen the diversity of it. What we have seen is they can be linear or circular, mostly circular in prokaryotes and linear in eukaryotes. And they can be single or many, and they can be some of the organisms can have very high number of chromosomes. Say some cells, specialized cells in our own body, like megakaryocytes, have about 128 copies of each chromosome. That's a huge number. Then we have gene density. Gene density is the number of genes per MB, if you want to talk it as a specific formula. MB, megabase pairs. Okay, and we have seen about we have learned a lot of terminology today. I think we will be using the terminology when we are talking when we are discussing in the class, or even when you're writing when I give a question also, it will be in those terminology. So although it could be jargon, but there is no easier way to, but we are also supposedly biology students, and I hope we will all get used to those terms and know the meaning of it. Often we lose interest in anything when we don't know the meaning of a particular word that we come across. Majority of the biology is learning the meaning of the word. When you split the words properly or the phrases properly and try to see what it means, then you can remember a lot of biology, or you can recollect a lot of biology. That itself is 75% success. Inter between genic genes. The region between two genes is intergenic region or intergenetic region. This is the intergenetic region here. And we will talk about pseudogenes and other things in the next class. If you have any questions, you may please ask.